Okay, well, thank you all for interested in joining this session. My purpose is today is like giving an introduction about what is AI and big data in cancer biology. So that would be like in the area of research that my group is currently beginning to be involved. We have a few years experience by now. So what I have put together is based on the experience that we have and some of the background knowledge that we picked up through the years as we work on this project. So the outlines for this lecture, I'm gonna first give you a simple overview. Oh, by the way, can you see my screen? Is everyone
Yes.
seeing my
Yes.
slides?
Yes, I
Yeah,
can
we
see.
can.
Okay, good. So first I'm going to give a simple overview of what is cancer biology. This is also a new subject for me. We are learning along the way as we investigate more and more about cancer. And then I'm going to talk about what are all the big data in cancer biology, where you can find it, what kind of data we have, okay? So today I'm going to just focus on the first two. And then later on, we're going to talk about what kind of AI, like machine learning, data mining algorithm that we may use to explore the big data to get some questions that's relevant to answer the cancer biology question. And I strongly encourage because I am myself use ChatGPT quite a lot. And just encourage you to use ChatGPT to learn. The key thing, however, is ask good question to ChatGPT. Then you can get good answer that's helpful to you. So always make sure you ask a good question. And the more you use ChatGPT, the better you can find that's really helpful in our task. Okay, so what is cancer? Um, so cancer is defined as just like cell that has underwent uncontrolled growth and spread of these abnormal cells. So it can grow on any tissue and organ, right? So this is also something the naming is new to me. Before I only know cancer, but as I learn more, I learned that they are defined cancer to carcinoma, sarcoma, leukemia, and a few more types. So these words is also very new to me, carcinoma. So carcinoma is the origin came from epithelial tissue, epithelial cells, okay? So they accounts about 80 to 90% of all cancer cases. Whereas leukemia and also lymphatic cancer, these are related in the blood. It's originated in the blood. And sarcoma is also a different kind, which is inside the bone. So as you learn about cancer, you probably would need to pick up more about cell biology and understand different cells in our human body, different organ. So it is a lot of learning. But these are like more common knowledge. Maybe some of you already knew these things. But in general, cancer is believed to be caused by mutations. The mutation can be inherited or acquired during the process, right? So when you talk about cancer, there is this so-called hallmarks of cancer. And when people talk about hallmarks of cancer, you can talk about this one. I grabbed one from this particular references, which focus more in like six of them. First of all, the cancer is able to sustain proliferative signaling and evade growth suppressor. The growth is like out of control, and they're activating invasion and metastasis. And yeah, and then they enable like replicative immortality. So in some sense, cancer cells live on forever. They don't die. So that's the problem. It's like escape the program the cell does. And another important thing is called inducing angiogenesis. Now, to me, all these words doesn't mean anything when you first saw it. So angiogenesis is one word that I really looking into it, what it means. So it means like it can grow the blood vessels inside the cancer so that the cancer can get nutrients more efficiently. So that's called angiogenesis. So it's basically like growing a tumor and has blood vessels grow inside. So it's like grow out of control. And then also another hallmark is resisting cell death. So these are like six of them, but people can talk more about them. So like if you take another paper and talk about hallmarks of cancer, like in this particular diagram, they have like 10 of them. So some additional known cancer hallmarks, it evades immune destruction, and it also promoting inflammation. And then the another one called it has genome instability and mutation. It has a lot of mutation and instability. And then it's deregulating cellular energetics. So yeah, so these are the cancer hallmarks that people associated with what is cancer. Okay, so I'm not a cancer biologist specialist, so my knowledge of cancer is also very limited, just reading from the literature and picking up these words and these discussion points. All right, so what is the big data in cancer biology? So that's the thing I want to share with you. So this big data in cancer biology, the collection of the data happened all around 2003. That's the year I moved to University of Memphis. And it happened after Human Genome Project completed. You guys heard of Human Genome Project? Have you before?
Yes, I have.
Okay. So I will show you the slides about Human Genome Project. So once the Human Genome Project completed, and we have a reference human genome, like a map of all of the base pair information on humans, um, then it began. We launched the NIH launched the Cancer Genome Atlas program called the TCGA. So you're going to hear me talk about TCGA all the time. Okay, that's launched in 2006. And it's a lot of study and a lot of data collected. And behind these data, it's really the thing that makes these data available is sequencing, especially the sequencing technology just went through some real big change during 2003 and last decade, very, very fast. So I'm going to show you later on about the low cost of sequencing, how it is like dramatically reduced from the year I joined till now. Okay. So, but before we begin, let me make sure we are on the same page. You know, we are human. And so we talk about genome. We know human has 23 pairs of chromosome, right? So this picture is really nice. 23 pairs of chromosomes, so total is 46, right? And yeah, but of course we know that this one and that one is same, right? So that's why we say 23 pair. Um, and the Human Genome Project the idea is actually to sequence all of the base pair in these 23 pair of chromosomes. So total is about 3 billion base pair, 3 times 10 to 9 base pair. So they want to sequence it and assemble them together into a reference map called Human Genome Map, HP. Okay, so the whole idea was want to have this map, all right? So let me yeah, okay. My next slide, I should talk about this Human Genome Project when it's began. I'll come back talk about this slide just a little bit later. Let me talk about this one. So the first Human Genome Project actually launched in 1990, uh, and they roughly completed, like they proposed the first human genome, um, the reference map about 2003. Initially, they planned for like 20 years, but later on they moved much faster, and it cost 3 billion US dollar. Okay. Now compared to the US government budget, I think right now $3 billion doesn't look that bad, right? Um, and so there's two important things. They want to know all of the sequence for sure, but they also want to map the gene. And so what is a gene and how gene mapping? So this kind of knowledge, you probably learn more in biology, and I'm not fully understanding how they figure out, but I know that the biologists worked hard together. So they were able to figure out how many genes total in the human chromosome and where are these genes located in our human genome. So basically, you just say they have worked out this detail. It's like a map, and every this little line is a gene. So you can actually go to this website, the Human Genome Data Viewer, and click each chromosome at each base pair. You will find information if it belongs to a gene or it doesn't belong to a gene, and what kind of gene name and what it is. So it is all there. So I strongly encourage you to click that link. That, in fact, I'm gonna click this link, see if I can take me there. Okay, it did go. All right. No, thanks. Did you guys see? So like this is chromosome 1, and these are the each base pair that you can click. And then the gene name like DPYD, these are all gene names. Okay. So yeah, that's how you can explore it. But of course, too, someone like us, like we first looked at it, like I'm not even sure how to make use of it. So that's these data are available there, but really we need to learn how to use them. Okay. Okay, so let me go back to my slides. All right, so then also I told you the human has 23 pairs of chromosome, right? And so the chromosome labeling is actually chromosome 1, 2, 3, 21, 22. And then the 23rd pair is related to sex, whether it's XX and XY. So the chromosome right now in the human genome, viewer is sex, chromosome X, chromosome Y, and there's additional one called mitochondria chromosome. Somehow there are some genes that's very specifically located in the mitochondria. And mitochondria is in the cell is like the powerhouse, right? They generate the energies. That's kind of powerhouse in our world, maybe you can think of that. Okay, so this has become available in 2003. And let me see. Yeah. And so when they finished this human genome project, they sequenced all of the base pair, like 3 billion base pair. At the end of the day, they found out that in the human genome, there's roughly about only 20,000 coding gene, meaning that that particular section of DNA is coded and you can find the protein product at the end. So there's only like 20,000 coding genes. And these 20,000 coding genes in terms of number of DNA base pair in the entire human genome is corresponding only about 1%. So it's a lot of the base pair is actually is not coding gene. Okay. So then let me give you one more slide. So then very quickly, people feel like, well, the whole NIH put so much effort to sequence all of the base pair in human that coding 3 billion base pair, right? But in the end, they found there's only 20,000 protein coding gene, and this is only has 1 to 2% of the 3 billion base pairs. Okay, so for the research purpose, instead of sequencing all of the 6 billion or 3 billion base pairs, we can focus on only 20 million base pairs that's associated with about 20,000 protein coding genes. And you don't have to code sequence everything, right? And so the rest is so-called a non-coding gene. I still remember in 2004, I was reading the news, and when they published the first human genome, first human genome, and they were saying there were so many DNA sequencing our human genome is non-coding. You don't see them in the final protein product. So you might call it like a junk genes. And so the biologists are still trying to figuring out what are the rest of the gene doing in our body that's there that doesn't have seems to codes for protein. So now they are understanding more and more that a lot of these non-coding genes probably is related to regulatory process, but a lot is still not known. It's just simply kind of like a dark matter in biology right now. Okay, so coming back to talk about sequencing. So you don't have to sequencing the whole thing. So there's another terminology that you will run into called the whole exome sequencing, the WES. This is only sequencing those coding genes by pulling only the DNA segments in the exon. Exon meaning like the region that will be expressed in terms of protein. So this will allow faster sequencing and allow researchers to identify mutations that's present in the exons. So you don't have to worry about all of those non-coding genes. So, but before I tell you more about sequencing technology change, no, I think I'm fine. So let's move on. And let me tell you a little bit about how the DNA sequencing technology changed so much from early 2000 to now. So the earliest sequencing technology, for some of you, you probably already know, is called the Sanger sequencing method. So Sanger sequencing method is a very traditional, the earliest sequencing method. You can learn that in molecular biology or any biology textbook, I believe. And so that happened in the technology became available maybe in the 1970s, 80s. And the Human Genome Project that launched in 1990 actually used the Sanger sequencing. This is called first generation sequencing, right? So in this type of sequencing, you every time you ran, you cannot run very, very long. You have to break the whole human genome DNA into fragments. And the fragment length that it can handle is only like 500 to 1,000 base pair. So you can imagine a very big challenge is that you have a huge DNA, 3 billion base pair, right? You chop to 500 to 1,000. So how many pieces you're going to have? You have millions of little pieces, right? So you do the sequence, and then after the sequence, you have to line them up correctly. So that's why it takes so much effort. It's like a group effort. So people have to figuring out like, okay, I'm going to chop this. And after I got this, how am I going to assemble them? So in the early days, these projects involve a lot of computer scientists that helping them to align these sequence after you did the experiment. Um, so this is what's happened in the bioinformatics. Actually, the earliest job is aligning. It is everything about aligning. You get a sequence, you align them. How do you align them? How do you align them efficiently? And after you align them, of course, you also think about, worry about how am I going to share the data with the people, right? So that's where the computer and technology come along together, has always been side by side, working on these problems together. All right, so this Sanger sequencing is relatively slow and expensive. And then the second generation sequencing happened in the early 2000s, the first decade in this century. And you probably will hear some name like Illumina. And later on, I will show you where you download the data. You will see the name Illumina HiSeq. So Illumina HiSeq technology, they developed a kind of like a high throughput by parallelizing the sequencing reaction. And so they still have to break it. They still only can sequencing pieces, like only 50 to 500 base pair fragments together, but they like parallelize. You can like one sequencing can run many fragments together. So it might make it much faster. And then later on, there's more sequencing technology that happened called third generation. And some of the instrument, you can hear the word PacBio is one that happens that's still in the market. Oxford Nanopore. So these sequencing technology is very different. It's called the third generation sequencing or next generation sequencing. And they can run the sequencing DNA very long. You don't have to break it. You like thread through the whole entire DNA into the instrument and then read the base pair off for you completely. So it makes the alignment job much easier because you are not breaking the whole DNA into very little chops, right? So I'm going to show you the next slides where I'm showing you the cost how much it costs to sequencing the DNA, the price change. In the 2000, the cost per megabase of DNA being sequenced is cost around $10,000. And then it's like, yeah, in the early 2001 to 2007, it didn't come down a lot, but it came down. And then look at this dramatic change. So by 2022, right now it costs how much? 0.01. This is dollar. So it's like what? It is so cheap now. So the technology of sequencing becomes so cheap, so affordable, everyone can sequence. And literally, you can sequence your own whole genome. It doesn't cost that much anymore. So that's where so much data can be generated because now we can literally sequencing everyone's whole genome. And so NIH begin to like launch a lot of program. So let's sequencing all of the patient with certain disease. Let's sequencing, yeah, they begin to like 1,000 genome, 1,000 people, and they want a different race, a different people group to be sequenced and store all of these data to become accessible. Okay, so a lot of data is generated because of this very fast dropping in the sequencing technology and becomes so accessible. Of course, the sequencing technology doesn't have to be used only in human. Sequencing technology is also heavily used to identify other living species like bacteria, virus, all of that. So the technology just changed the kind of like how we interact the world dramatically. Okay. So um, before I talk about there are different type of sequencing approach, I wanted to bring into another slide over here. This is the basic biology, right? So if you have taken biology, you kind of know that inside our cell, the genetic information was stored in the DNA, in the nucleus, right? And then in order for the information to be eventually translated into protein, so it goes through DNA, needs to be transcribed to RNA. And this process uses RNA polymerase. It transcribes DNA to RNA, right? So that's the step that happens inside the nucleus. Okay. And so the RNA, when it first transcribed, is called a pre-mRNA. It's not a mature messenger RNA yet. All right. And it will go through some post-transcriptional modification. And these modifications include like capping, polyadenylation, like adding the poly A tail on the messenger RNA. And there's also different splicing and RNA editing. And then it become a mature messenger RNA and then exported into the cytoplasm. Okay. And then in the cytoplasma, the messenger RNA is taken up by the ribosome. So the ribosome will read the code on the messenger RNA, and the ribosome will translate the messenger RNA to protein, right? Ribosome is the main machine that translates the messenger RNA into coding protein. So I give a picture of here, the ribosome. And also one more thing you need to know that for the mature messenger RNA, this is a picture of mature messenger RNA. No, I don't want to annotate, sorry. This is the picture of mature messenger RNA. So it has a poly A tail, right? And this is the coding region, and then a little bit has a 5' untranslated region and 3' untranslated region and the cap. Okay, so that's a mature messenger RNA. And then this coding region, you can find the corresponding protein in the cell. So that's how, all these information fit together, right? And then protein are the one that do the most cellular activities inside cell because protein is the one that work around all of the things inside the cell. Okay. Let me close this one. All right. So with that in your mind, now let's look at sequencing technology, instrument-wise, you have different instrument, but it's become very cheap. However, you can also adapt your sequencing into different ways. So here's some acronym that you will hear. The first one is called whole genome sequencing, and the abbreviation is WGS, whole genome sequencing. In case of the human, this is referring to that we sequencing all 3 billion base pairs in human, okay, including both the coding and the non-coding region. Yeah, what do you mean coding? Well, the coding one is the one that you can find that piece of the DNA has the final product, the protein. So that's the coding gene, right? Non-coding region is whatever in between that you don't find the final product in terms of protein. So you can do whole genome sequencing. And when you do whole genome sequencing, of course, you can find mutations both in the coding region and non-coding region. But on the other hand, you can also do so-called whole exome sequencing. And so the abbreviation is WES. So in these cases, you only pull down the coding region. That's about only 1% of the whole human genome, about 20,000 genes. And then sequencing is much faster, much cheaper to do that. And you focus on mutations in the genes that has the final product of protein. So you can kind of figure out what are the mutation in the protein you can find. Okay, so this is whole exome sequencing, WES. And then there's another technology called RNA sequencing. So the abbreviation we always say is RNA-seq. So in this technology, you don't sequencing the DNA anymore. You actually pouring down the messenger RNA from the cell, and you're only sequencing the messenger RNA. And so one of the trick to pouring down the messenger RNA, remember I said the mature messenger RNA has a poly A tail, right? So you can do some tricks by pulling only the poly A tail, some kind of tag that attached to only the poly A tail. So all of the messenger RNA with the poly A tail being pulled down, okay? And then these messenger RNA is being sequenced. Yeah. So and when it comes to the how exactly sequencing, that's molecular biology take care of that. You don't have to worry too much. But basically, you take the messenger RNA, you reverse transcriptase to the DNA, and you amplify and you put it in the cell to be sequenced. So RNA sequencing is a way that we can now actually measure how much that particular gene, its messenger RNA, are present in the cell. So this information is used to correlate the gene expression, because gene expression somewhat is related to the messenger RNA concentration inside the cell. Now, it's not exactly the protein expression level, but it is okay. So most time people will say gene expression. They are actually referring the gene expression measured by RNA-seq, not directly measured by the protein product, okay? But it has become a common way to phrase it, say gene expression measured by RNA-seq. There's another technology that is very new in only like recent years. It become accessible to most of the experimentalists. It's called single cell RNA sequencing, right? So this technology differ from the RNA sequencing is that in this, the bulk RNA sequencing, you just have certain sample, right? You have many cells. You don't care about what's inside the cell. You don't identify the cell. You homogenize them. You're pulling out only the messenger RNA, and then you sequence them, and then you just count, okay, all this messenger RNA, what gene it is, and you line them up on the human genome. You say, okay, this is this gene, this is that gene. So you have an idea out of that whole bulk tissue, that particular gene has how many messenger RNA, right? And then a single cell RNA sequencing, how does that differ? What it does is that inside this sample, each cell, it has a barcode. And so when you're sequencing the messenger RNA, not only you now align that particular messenger RNA to a gene, it also carry a barcode that tells which cell it belongs to. So now you can actually know each individual cell how much that particular gene is expressed. So this technology creates more data, more, how should I say, in-depth data, which you can observe the gene expression inside your tissue, not as a whole sample, but embedded in individual cell. So it can reveal the expression at the single cell level. And so these data reveal cellular heterogeneity, and it can tell what types of cell you have. So a lot of bioinformatics are right now working on these technology is to identify the type of cell you have inside your sample and how you sort them, how you identify them, and then understand inside that particular cell what gene is expressed. So this has been used to identify like cancer stem cell and to characterize the tumor microenvironment, right? There's more. There's a few more on sequencing technology. Another one called chip sequencing. Chip. Chip is referring so-called chromatin immunoprecipitation assay. What it does is like it can pull down part of the DNA that has protein bound on it, and it used some chemistry to cross-link it, and you pour on the protein that with the DNA bound together. That's called chip. And so then they take this DNA that has been poured on in this way to sequence it. So this approach allows you to sequence only the section of DNA that has transcription factors or histone, that these protein that binds on DNA, to allow you to pull down only that section of DNA. So this allows you to understand the epigenetic regulation on the gene expression. Yeah. Another technology that you will hear is called ATAC sequencing. ATAC is a word that talks about is the initial stands for assay for transposon accessible chromatin, right? So what it does, it is, yeah, it's kind of, in my mind, it's just like it is just looking at the whole chromatin and the part that is accessible is not bound, not like closed up. So only that part of the chromatin is being pulled down. So it will reveal how the chromatin is being open or reconstructed during the process. And then you can potentially understand the gene regulation better. So I know a lot of biologists is really try hard to understand the gene regulation, because you knowing the gene, but it's the gene regulation process that actually impact how the cell behaves, right? Another sequencing technology, to me, it is called bisulfite sequencing. So this is actually very related to the chemistry. It is using the bisulfite that's bind on DNA, and I forgot the details. And then they can like differentiate. Some of the DNA has being methylated. Some of the DNA is not methylated. And so this technology is able to identify those methylation pattern on the DNA. And so it's called the DNA methylation. So these data will report you on the DNA, the genome, which side you have methylation. So this is DNA methylation pattern. So then DNA methylation is also related to epigenetic control on the gene regulation. And it is related to the tumor genesis, and the people pay quite a lot of attention to the DNA methylation pattern. Okay. Do you have any? Sorry, I just saw a chat. I want to make sure nobody. All right, so let's go on to talk about the Cancer Genome Atlas Project. So if you Google it, say the Cancer Genome Atlas Project, you will be led into the TCGA website, right? And this is stated in NIH, the Cancer Genome Atlas, TCGA. It's a landmark cancer genomics program, and their goal is to molecular molecularly characterize over 20,000 primary cancer and match the normal sample and spanning 33 cancer types. And so this is a joint effort between National Cancer Institute and National Human Genome Research Institute that began 2006, right? And so over the next 12 year, a dozen year, TCGA generated over 2.5 petabytes of genomic, epigenomic, transcriptomic, and proteomic data. So these data has been very valuable in research. And so they will remain publicly available for anyone in the research community to use. So the data is always there and it is made available for the research community. And it's really hoping that everyone who are interested in biomedical field of research can make use of that. So this is in the cancer. And then you can imagine all the disease now has those various kinds of data that made publicly available. So if you're interested in any sense of biomedical field, you really need to know what's the data out there and how we can make use of them. Okay. I draw this the Cancer Genome Atlas project, the main event based on that website. You actually can go to the TCGA website. They will show you the important timeline in how this TCGA project is going on. So they've launched initially is just a pilot program. And then they have, I forgot, BRCA is the breast cancer. This is the one I'm kind of focused on. Launched in 2012, maybe. And then 2013, pan cancer analysis group formed. That's like they focus all kinds of cancer together. And then the data they figure out that we have to share the data. So a lot of computer scientists come together, working together. They launched the so-called Genomic Data Common, GDC. That's where all of data are shared, loaded, and distributed. Okay. So I'm gonna take you to this called GDC, Genomic Data Common. So that's the website that the research has using to load the data, to share the data. So there's a huge amount of data. You can go there and find it. This is their picture, which showing the types of cancer that they have data on. The breast cancer, the ovarian cancer, colon cancer, kidney cancer, lung, and so on. And for each type of cancer, they have different types of data that's loaded there. The clinical data, which is just referring, what type of person that is sample from, how old it is, and whatever the treatment he has received, how long he has lived, and so on. So that's the clinical data. And then the sample from that person will be sequenced. So there's mutational pattern. These are just different samples, different genes that person has sequenced, where all the gene expression, so on. So there's mutation, copy number variation, gene expression, DNA methylation, microRNA, and RPPA reverse phase protein array. So these are the data that's all available for that particular cancer types. And then, of course, there's another higher level of data analysis. Not only you looking at these data in a two-dimensional table, then you worry about gene-gene interaction and pathway. So this is the data that generated based on those huge data sets to generate a pathway, that understanding how gene-gene interaction. Okay, so I am going to tell you, well, even though the GDC is where you share the data, but I will tell you that there is a very good website that is much easier for you to download the data. That is the Xenon browser Xenon browser. Xenon browser has a lot of data. So I'm going to click this. Let me see if I close. How do I close that? Oh, sorry, that's not what I wanted. I'm not very good at using this. Okay. let me see if I can I close. I'm in the laser mode. I want to get rid of the laser mode. How do I get rid of that? Highlighter. No. Um, hmm, maybe select. No. Draw. Oh no, what is that? Pen. Um,
When you repressed it, did it allow you to be out of the laser mode?
when I press
Like,
here,
press it twice. What
press
or like
this, like that. No, see,
no, like how you were just in there, you switched it to the pen mode.
right.
Click
So
on the like
I mean here, right?
yes. Now click on the pen mode again.
Pen mode.
Now
Oh,
you
yeah, that worked.
there you go.
Okay. Thank you, Dylan. Okay, so if you click this one and then you go there, this is the Xenon brother. And I go to the data sets directly. If you somehow don't remember where it is, you can Google, you say Xenon brother, okay? You can come here. And so this is the first page. And it does give you a lot of tool how to view the data, but I did not find viewing the data on the browsers that helpful. What we do is always go to the data sets and download the data and we manipulate data ourselves. So you go to data sets and then you can see that there's a lot of different, right? So these data sets are arranged by the study, like what type of study. So let me show you, for example, um, MAP 500. Right. So for example, I say a lot about TCGA, right? So TCGA, colon cancer. So that will be the Cancer Genome Atlas launched that study focused on the colon cancer. And so that defines that all of these sample is from the patient that with colon cancer. So you click this link and then you will see a lot of data that's here. So I found this page is very helpful. It's just organized very well. So you will find, okay, the type of data they will offer you is either the first one is related to copy number variations. And I'll tell you a little bit about that concept shortly. And then also tell you DNA methylation, right? Which I mentioned. If you sequence, you found the DNA methylation spot, and it's important. The bioinformatic, biostatistician, they are analyzing DNA methylation patterns in the cancer patient. And then exon expression for RNA-seq, but it also had gene expression RNA-seq. Now, I still did not figure out what's the difference between this one and that one. But in general, we use the gene expression RNA-seq data, and we use this aluminum high-seq. So this is aluminum high-seq. N equals 329. That means like they have 329 patients on TCGA hub, right? So this is the one that we use normally. And so these data is actually, they did RNA sequencing. The bioinformatics people already take the raw data out of the instrument and aligned on the genome. And the data is come out is only that after you align each particular gene, it reports how many counts of messenger RNA you have. So it giving you the data is a table where these are just gene name, and then each gene name, how many counts you have, okay? So that's the gene expression data. So DNA methylation data is slightly different. It doesn't comes into particular gene. It is actually where the methylation sites you have on the genome. So yeah. And okay, and what else? And they are mutation as well. And the phenotype data. So okay, so this one, the curated survival data, this is the one that actually I told you like called clinical data, right? That is equivalent to the clinical data in your in your head. It tracks the patient information, like whether this patient age or what kind of is it female or male, and then what race it is, and also like how long lived, what kind of treatment has. So that's where it is. So it is curated survival data. Is they already like process the raw medical record and make it much easier for us than non-medical field person to look at data and understand and use it. So for us, for a lot of analysis, we use the so-called overall survival or progress-free PFI or DFI, DSS. These indicator, I can't even recall. These indicator that shows like how long that patient has lived. So this is an important data that you need to use for data analysis purpose. Okay, I'm gonna go back to my slides. All right. So for each of these data sets I showed you, you just need to know that they are organized by the study. So I told you a few examples I walk you through. The BRCA is the breast cancer, prostate cancer, the initial is PRAD, and pancreatic cancer is PAAD. Okay, so these data, this is I define the type of tissue, the type of patient, the type of tumor they have. And then under each study, you have different data sets I walked you through earlier, right? Copy number variation, DNA methylation, gene expression by RNA-seq data, survival data, and protein expression by RPPA approach. Okay, so when you think about data, you just think of data in terms table. Everything is a table. So when you're dealing with data, important thing is a table. So what's table? Important thing is what is the table? Each row label, which column label, right? So I kind of drag this table out of the web. So for example, in the RNA-seq data, so each row is different sample, like different patient, right? Then each column is actually different gene. And then the value here, table cell value, the value is the gene expression level in the sample one and that gene one, okay? And then of course, sometimes you may have additional row, additional column that will associate some other not necessary gene expression. Like in this case, it's just associated that diagnosis is a normal or it's a tumor. Okay, so when you're dealing with these data analysis, you're gonna just continue to think of all the data is coming in the table. So you just become very used to think of this in this way. It's a table and what's the data, what's in the row, what's in the column, and how am I gonna process it. And we're gonna use, yeah, I talked about this already, so I don't need to tell you much. The gene expression by RNA-seq is the one we focus heavily on. So this is measured by messenger RNA transcripts captured and aligned. And the gene names, each gene name, So the biologists really work hard. They have a systematic way of naming all the genes. And they do provide a file called ID gene mapping probe name. And then you use that to know what's the name of the gene, okay? So we will talk about in our research, we'll talk about integrin gene, we'll talk about CEA, CAM genes. So you can type the name, but you need to make sure like a gene and the name are mapped together. And then we can investigate the gene expression for these genes. So you will be trained to use the Python code to read the data and manipulate data in Jupyter Notebook, okay? And my graduate student, Dylan, I can call you my graduate student now, right? Vish is also a graduate student. They are more advanced, definitely. They are learning the Jupyter Notebook. They're pretty good on it. So I will like work with them to help you to learn these. So we will be able to explore gene expression, gene mutation in cancer using the Python code by download these data and look at these data on your own, okay? So the training plan is that I will want you to learn some Linux command and install Jupyter Notebook on your personal computer. I'm a big Linux user and I might be wrong, but I feel like this world where Jupyter Notebook installation, etc., actually has a lot backend is related to the Linux command. If you understand these things better, you understand the whole the world of computer world better. Uh, Maybe I'm biased. Okay, and
Chuck,
another what Dylan?
I was gonna say you do know that Big Blue has the GUI interface live now too,
Oh, okay.
so they might not need it as much.
But I still prefer you guys learn the Linux command because it really Linux command makes you control the computer, like file name, etc. Um, yeah, or yeah, maybe I don't know. And then I will ask you to create a GitHub personal blog page and also begin to use GitHub repository. So that's the way we can exchange code. You can share with me and I can share with you, and we can check the code, how it working. And you will run Jupyter Notebook to learn Python coding through the Jupyter Notebook. Okay, so this is really the goal. And so whenever it's related to the computer training, it's best done in person.